I once again welcome all the participants for the technical session 2. Now I request Dr. S. Nandakumar to introduce the speaker, Dr. S. Kavimani, Professor, Head, Department of Pharmacology, Mother Theresa Postgraduate and Research Institute of Health Sciences, Puducherry.
Very good morning. I welcome all the participants to the second technical session. Though the speaker of today's webinar does not require any introductions among the pharmacy professionals, I take the privilege and honor to highlight the overview of his career. Dr. S. Kavimani graduated his UG degree from Madras Medical College, Chennai. He received his post-graduation as well as PhD from Jadavpur University, Kolkata. Dr. Kavimani has 27 years of teaching and research experience. He has guided 42 PG students and 7 PhDs. He has published more than 230 publications in national and international journals. He has published 2 books and has written a book chapter on Elsevier. Dr. S. Kavimani is an expert committee member for Pharmacy Council of India, AICTE, and NBA. He is a Board of Studies member in Puducherry University, MS University, JNTU Kakinada, and Annamalai University. He is a visiting faculty for Puducherry University and MS University. He is presently serving as the Professor and Head of the Theresa Postgraduate and Research Institute of Health Sciences. I welcome Dr. to speak on discovery of drugs, doctrine of signature to pharmacogenomics. Welcome you, sir.
Thank you for the introduction, Dr. Nandakumar. Am I audible, Nandakumar?
Yes, sir, you are audible, sir.
Yes. Can I share my screen?
Yes, sir, please. Yes, sir. The screen is visible, sir.
Yes,
so kindly keep it in the full screen mode, sir.
Oh, okay. Very good morning to all the participants. I would like to thank the management, the principal, and staff members of Faculty of Pharmacy, Dr.M.G.R. Educational and Research Institute, for giving me the opportunity to deliver a lecture in the E-Faculty Development Program. Dear participants, good morning to all. Today I am going to present a lecture in the title of Discovery of Drugs from Doctrine of Signature to Pharmacogenomics. The content of my lecture is what do you mean by Doctrine of Signature and what will be the traditional methods of drug discovery that is followed by the modern methods of drug discovery by using the genomics, drug in case of COVID-19 and serendipity, and that is followed by the digibiotics. Now I would like to discuss about the doctrine of signature. Now what do you mean by doctrine of signature? By observing the plant, we can determine from the color of the flowers, root, or shape of the leaves, the place of growing and other signatures, what the plant purpose was in God plant. I would like to give some examples for that. When there is a will, definitely there will be a way. When there is a disease, definitely there will be a remedy. If you are in higher altitude, if you go to higher altitude, definitely there will be scarcity of oxygen and there may be possibilities of development of respiratory disorders, especially asthma in susceptible individuals. The ephedrine is an alkaloid which is obtained from the Ephedra sinica, which grows only at higher altitude of Meghalaya and Sikkim in case of India. This is a very good remedy in Ayurveda for respiratory disorders. Now, this is the plant is called Catharanthus roseus. are vinca alkaloids from which vincristine and vinplastine have been isolated. And this is one of the ingredient of MOP regimen, which is used for various type of cancer, especially Hodgkin lymphoma, which is nothing but mecloratamine, oncovin, procarbazine, and prednisolone. The oncovin is nothing but onco means cancer, vin means vincristine and vinplastine. So these alkaloids, this plant, almost we can find all the medicinal gardens of the pharmacy colleges. And you will find two varieties of flowers. One is white colored variety, another one is pink colored variety. So the vincristine and vinplastine, which is isolated from the white colored varieties, are more effective in case of lymphoma, ascites lymphoma. You know, ascites is white in color, whereas the vincristine and vinplastine, which is obtained from the pink colored varieties, are more useful in the treatment of leukemia. So from the color, we can say what will be its use in cancer. You can see the sliced carrot, right? It looks like our eye. The pigment which is present in the eye is also present in the carrot. The rod and cones, which is responsible for the vision, will be maintained by these pigments, that is called carotenoids, which is highly rich in carrot. Now, tomato, the most important antioxidant isolated from the tomato is lycopene. Lycopene is very much effective against the chronic mild stress-induced depression and also isoprenaline-induced cardiotoxicity. The various researchers have explained, proved that the lycopene is having very good antioxidant activity against isoprenaline-induced cardiotoxicity and also chronic mild stress-induced depression. Here you can see the tomato. These tomato have 4 chambers. Our heart is also have 4 chambers. So North Carolina researchers have shown an increase in tomato products may reduce the risk of congestive cardiac failure. We take the example of walnut. The walnut looks like a little brain, right? Our brain is regulated by various neurotransmitters. Some of the neurotransmitters are excitatory and some of the neurotransmitters are inhibitory. Right. So walnut is also having 3 dozen neurotransmitters, which is responsible for the maintenance of our brain function. The wrinkles which is present in the walnut is also present in the brain. So based on the what you call the shape, the morphology, we can say the walnut is very effective against the maintenance of neurotransmitter level in the brain. The kidney beans, it looks like kidney. You can see the hilum of kidney beans and also the hilus of the kidney. The person who is under the treatment of therapy of aminoglycosidal antibiotics, especially streptomycin, kanamycin, there may be chances of nephrotoxicity. So in that circumstances, supplementation of the kidney beans with the aminoglycosidal antibiotics may have some kind of protective action in those patients. Next is the rhubarb. The rhubarb, it looks like just like our bones. Not only the morphology, the content of the rhubarb, it contains 23% of the sodium, and the bones also contains 23% of the sodium. Like calcium, sodium is also responsible for the maintenance of the functions of our bones, and also the sodium can replenish the skeletal needs of our body. Now take orange. It looks like our mammary glands of female. The squeezing of this orange leads to the juice. Like that, the stimulation of the lactiferous tubules of the mammary glands leads to secretion of the milk. And orange is lactocoque, which increases the secretion of the milk, and also they assist the health of the breast and the movement of the lymph in and out of the breast. Take onion. Onion have a pungent principle which can increases the secretions of the tear. It is one of the defense mechanisms. By increasing the tear secretion, we can send out the cell debris or metabolic waste. So this onion looks like our cell. You can see the picture. Next, I would like to discuss about the peanut. Peanut is almost available in pairs. Our testis is also in pair. One of the most important amino acid which is responsible for this spermatogenesis is arginine, right? So the peanut contains enough amount of arginine that can increases the spermatogenesis capacity, so that it can be used for the treatment of loss of libido, loss of sexual libido. And this picture shows the aerial roots of banyan tree. Here we are mimicking the aerial roots of banyan tree with our hair. As you know that this is a very good hair tonic. Next, Garilla. Garilla is nothing but Mogana SRA, which is very much useful for the treatment of diabetes mellitus. Here I would like to discuss two concepts. One is the ripe fruits of Mogana SRA looks like our pancreas. The second one is any bitter principle can be nullified by the sweet. Like that, any sweet principle can be nullified by the bitter. So that in case of increased glucose level in our blood, that can be neutralized, that can be reduced by taking a more amount of Garilla. Next, bragmi. Bragmi looks like our brain. You can see the structure, right? They are very good memory enhancer. Now, distalis. It is a first plant used for the treatment of congestive cardiac failure. The blood colored spot which is present in the distalis, it is nowhere present. No flowers contains this kind of blood colored spot, and also the leaf of the distalis purpurea, it looks like our heart, so that it is used for the treatment of congestive cardiac failure. Chelidonium. The juice of these particular flowers are used for treatment of hepatitis or jaundice. Since you know, jaundice is due to increased secretion of the bile, which is yellow in color, so that the chelidonium juice can be used for the treatment of our jaundice. It is called spleen wort. It looks like spleen. It looks like spleen. As you know that.
Third class, two class. Okay.
Please mute your mic. Participants, please mute your mic.
Daddy,
Dear participants, please mute your mic.
Daddy,
Right, okay, thank you. It looks like a spleen. The RBC destruction takes place in spleen. One of the reasons for hemolytic anemia is overactivity of the spleen, so that can be controlled by administration of this spleen wort. This is liver wort, right? The lobes of liver. The liver wort have various lobes, which looks like our liver lobes, so that it can be used for the treatment of liver-related problems. This is called tooth wort. Tooth wort. This floss looks like our molar. Usually, you will find dental caries mostly in the molar tooth. So, looks like our claw, and the juice of this flower can also be kept in the tooth caries, and it have very good antibacterial activity. Right. This is lousewort, which looks like lice. So that the most important ingredient of anti-lice shampoo or anti-dandruff shampoo is nothing but the lousewort. So far, what I have discussed is doctrine of signature. So now we can able to say what is meant by doctrine of signature. It is nothing but by observing the plant or parts of the plant, morphological character, or the color, or the shape, or where it grows. Like by observing those characters, we can able to predict the possible therapeutic efficacy of those medicinal plants. Right. And this kind of information about the plants, you can get it from the Wealth of India, Ayurvedic medicinal plants, Ayurvedic pharmacopoeia plant drugs, Ayurvedic pharmacopoeia of India, and Tamil Nadu Carnatics, and Encyclopedia of Indian medicinal plants. So these are all the available sources in the form of book to get the information about the medicinal plants. Right. Now, what is meant by traditional methods of drug discovery? Now, I said the plants derived materials. So far, I have discussed that. The salicylic acid, which is isolated from the willow tree, right? The salicylic acid, dear participants, please mute your mic. There are a lot of disturbances, please mute your mic. So, this is a structure of salicylic acid. As we know that salicylic acid cannot be given by oral route, it have the high incidences of the ulcer. Salicylic acid along with the benzoic acid is used in the form of ointment. So what we have done is we have acetylated the salicylic acid to get the acetyl salicylic acid. It's a simple acetylation reaction so that we got acetyl salicylic acid. Acetyl salicylic acid is not a very good anti-inflammatory drug. It is only an analgesic at a dose of 300 milligram. So that there is substitution of the propyl group. So it is ibuprofen. Ibuprofen is a very good anti-inflammatory activity, but it have the incidences of gastrointestinal side effect, that is gastrointestinal hemorrhage. So that why the gastrointestinal side effect? Because it inhibits both cyclooxygenase 1 as well as cyclooxygenase 2. So instead of that, specific COX-2 inhibitor like etorecoxib, proficoxib, and celecoxib have been developed, which is completely devoid of the gastrointestinal side effect. Right. This is that is synthetic, semi-synthetic, and natural drugs obtained from the plant origin. Right now, we can see this the synthesis of salicylic acid either from the salicin and chemical substance, or from the methyl salicylate, either from the salicin or from the methyl salicylate. Methyl salicylate on hydrolysis or salicin on hydrolysis produces salicylic acid. Salicylic acid on acetylation produces acetyl salicylic acid. Now, this was first introduced and marketed by the Bayer in German during 1897. Now, we know the age of the aspirin. Now, an apple per day keeps the doctors away. It is a proverb. But now, an aspirin per day keeps all the cardiovascular complications away. The dose is 60 milligram or 75 milligram because aspirin have antiplatelet activity. As you know that platelet aggregation is one of the complications in the cardiovascular problems. Right. Now, drug discovery from the marine organism. As you know that marines are highly rich with fauna and flora. They have various microorganisms, various organisms called tunicate, mollusk, cyanobacteria, sponges, fish, and bone snail. Now, many number of drugs have been identified and isolated from the marine organism, especially for the treatment of cancer, for various infectious diseases. For example, trapectidine, Trastuzumab, these are all used for the treatment of soft tissue cancer and also malignant lymphoma. Eribulin mesylate, which is isolated from the sponge, can able to act as a mitotic poison, interfere with the microtubules, thereby it is used for the treatment of metastatic breast cancer. And omega-3 fatty acids, you know that the statistics says that the non-vegetarians are less prone for the development of congestive cardiac failure or myocardial arrest when compared with the pure vegetarian. That means pure vegetarians are highly prone for the development of cardiovascular complications because omega-3 fatty acids, which is richest source available in the fish, which is an antioxidant. So taking marine-derived product may protect our heart from the cardiovascular complications, and also it is reported that it can able to reduce the triglycerides level in the blood. There is most important analgesics, giconotide, which is used for the treatment of severe and chronic pain. Vidarabine and cytarabine, these two antibiotics can able to inhibit the DNA polymerase, thereby they are all used in the treatment of herpes virus infection or for the treatment of leukemia. There are many number of drugs are obtained from the marine. So I said siconoside, they block N-type voltage gated calcium channel. As you know that calcium channels are in almost in all cardiovascular system like angina pectoris, cardiac arrhythmias, and hypertension. There is the use of calcium channel blockers. I said siconoside is a non-opioid analgesic, but can be useful for the treatment of moderate to severe pain. So if you use opioid analgesics for severe and moderate pain, there may be chances of addiction. But by taking this kind of medicine, we can escape from the addiction liability. At the same time, for cancer-induced pain, it will be useful. Now, cephalosporin, it is a major group of antibiotics which is obtained from the marine. I said omega-3 fatty acids. Right. Now, is it possible to obtain the drugs from the human source? Yes, it is possible. I will ask two questions. What is the confirmatory test for pregnancy, and what is the mechanism of the Gravidex test? Gravidex test is the confirmatory test for pregnancy. What is the mechanism? The first urine of pregnant woman, if positive for the human chorionic gonadotrophin, then they are said to be pregnant. Right. The second question is why pregnant women are not getting menstruation. Human chorionic gonadotrophin, which is secreted from the placental unit, which can able to maintain estrogen and progesterone level throughout the pregnancy. As we know that if there is no decline of estrogen and progesterone, there won't be chances of menstruation. Right. So this human chorionic gonadotrophin is isolated from the urines of pregnant women, and it is very much useful for the induction of ovulation. In case of infertile women, if the infertility is due to lack of ovulation, we have to give human chorionic gonadotrophin. The next drugs obtained from the human is monotrophin, which is obtained from the most post-menopausal women. This is the richest source of follicular stimulating hormone and luteinizing hormone. Again, the monotrophin is also the ovulation inducer. As you know that from human we have isolated the insulin, and urokinase, which is having antithrombolytic activity, which is obtained from the human kidney cells, and also recombinant growth hormone, which is also obtained from the human. So these are the drugs obtained from the human. Right. Now, what is meant by modern methods of drug discovery? So far, what I have discussed is a doctrine of signature and the traditional methods of drug discovery. Right. Now, what is modern methods of drug discovery and how it is different? Is nothing but new and improved methods of drug discovery. Here, the drug discovery process begins with a disease rather than a treatment. You know that if you take the topic of antihypertensive drugs, you will find almost 60 to 70 different types of drugs, which belongs to 11 to 12 subclassification. It is angiotensin, AC inhibitors, angiotensin antagonists, all these things. Now, how the physician select a particular drug for that particular patient is also depends upon the pathophysiology of that particular disease. So now use disease model to pinpoint relevant genetic and biological component which can be a better possible drug targets. Right. Now, how we are going to choose a disease for your drug discovery process? Right. This is the disorders of third world countries, these underdeveloped countries like Kwashiorkor and marasmus. So these kind of diseases prevailing only in third world countries or the underdeveloped countries where they don't have, this is a nutritional disorder, they don't have money to buy to get the food. So in that circumstances, if you can able to develop a drug for Kwashiorkor and marasmus, definitely the marketing potential will be very low. So no pharmaceutical companies will come forward to develop a drugs for the Kwashiorkor and the marasmus. Whereas the disease which occurs in first world countries, that means developed countries, for example, cancer, cardiovascular disorders, depression, diabetes, migraine, obesity, etc. As per the WHO, more prevailing disorders of the world is three. One is hypertension, second is atherosclerosis, and third one is diabetes mellitus. So, if you can able to develop the drugs, even though many number of drugs are available for these disorders, if you can able to develop a drug for this kind of disorders, definitely there will be a marketing potential. Right. Now, we will discuss about the drug discovery process for cancer. Right. I said it is a more prevailing disorders. So, if you see the pathophysiology of tumor development, it belongs to 6 categories. One is sustaining the proliferative signaling. That means there will be uncontrolled proliferation of the cells which forms the neoplasm. That is nothing but newer growth. The second is evading growth suppressor. The third one is activating invasion and metastasis. Right? What do you mean by metastasis? The tumor can able to spread from one region to another region. That is called invasion and metastasis. Okay. We are going to enabling the replicative immortality or inducing angiogenesis is also one of the pathophysiology of cancer. The last one is resisting the cell death. So I can able to discuss some 2 points regarding the drug development process of cancer. The first one is, if you see the difference between the normal cells and the cancer cell, right? See, this is the telomere chromosomes. You know, at the end of the chromosomes, you will find the telomere. Right. For each and every cell division, replication, the size of the telomere is getting reduced slowly. Right. Now, at one stage, there will be apoptosis. So, apoptosis is nothing but programmed cell death. it is a natural killing of cells. For example, the lifespan of the RBC is 120 days. Within 120 days, the RBC will die. That is called the apoptosis. Whereas, in case of abnormality in the cell growth, we can see even after so many replications, the size of the telomere is remain consistent, so that there is no question of apoptosis. If there are no apoptosis, there will be further proliferation of the cell that leads to uncontrolled proliferation, which we are saying that the cancer. Right. Now, what do you have to do? We have to induce apoptosis in cancer patients. How we can induce apoptosis? How we can develop the drugs for cancer means we have to develop the apoptosis promoter. That means for the apoptosis, what are all the genes are responsible? For example, BAX or BCL or caspase. So now the research is going on for the induction of the apoptosis either by the extracellular stimuli like caspase or by the intracellular stimuli like procaspase 9 or APAP1. This is the recent advancement in the cancer discovery process. The second one is, you can see here, it is angiogenesis. What is angiogenesis? Suppose if newer and newer cells are getting formed in our body, that is the development of tumor. For the newly formed cells, we have to supply the nutrient. So if you want to supply the nutrient, definitely there should be a formation of the new blood vessels. For the formation of the new blood vessels, we require three factors. One is vascular endothelial growth factor. This is vascular endothelial growth factor. This is platelet derived growth factor. This is fibroblast growth factor. So now this growth factor, fibroblast growth factor or vascular endothelial growth factor or platelet derived growth factor can be able to combine with their receptors, thereby it increases the formation of the newer blood vessel. So now what we have to do? We have to suppress the angiogenesis. If you suppress the angiogenesis, if you inhibit the angiogenesis, definitely there won't be supply of nutrient or blood for the newly formed cells. So automatically there will be cytotoxic activity. So these endogenous inhibitors of angiogenesis were either a matrix derived or the non-matrix derived. And these are all few examples. Just I would like to read arrestin, canstatin, fibrillin, endostatin, thrombospandin 1 and 2. These are all matrix derived, whereas in non-matrix derived, troponin 1 and vasostatin, angiostatin, etc. So this is about the angiogenesis inhibitors. Right. Next one is sustaining proliferative signaling. What do you mean by sustaining proliferative signaling? There are the intracellular signal. Because of the signal, like second messengers, because of the intracellular signaling, there will be what you call the proliferation of the cell. Actually, normal cells rely on positive growth from other cells, whereas cancer cell can reduce their dependence on growth signal. How? By production of their own extracellular growth factor. The cancer cell can be able to produce their own extracellular growth factor or the overexpression of the growth factor receptors or by alterations to intracellular components of signaling pathway. Now I would like to explain, I would like to give one example. The example is Herceptin. Her means she, septin means inhibition. It is a drug used for the treatment of breast carcinoma. See, the irregularity in menstruation or over menstruations is due to the fibroids in the uterus or in the cyst in the ovary or in the cancer in the breast. Right. Now, the drugs which is developed for this mechanism is Trastuzumab. You know what is it? Trastuzumab. So this is the normal breast tissue cell, and this is the HER2 gene. Right. The HER2 gene is normally expressed in normal patients, normal subjects, whereas in case of breast carcinoma, the HER2 gene is overexpressed. You can see here a number of HER2 gene. When the gene is overexpressed, automatically there will be proliferation of the cell that leads to breast carcinoma. Now, Herceptin, which is a monoclonal antibody, which is denoted in yellow color, it is an antibody. This Herceptin or Trastuzumab can able to combine with this HER2 gene, that is the surface receptor. Thereby, it reduces the progression of the cell growth. Right. Now, these are some other examples of drugs developed by this mechanism, and most of drugs are used for the treatment of cancer. Some of them, you know, the letrozole, tamoxifen. Tamoxifen is an estrogen receptor antagonist. It was the drug of choice once for the treatment of breast cancer, but now this is replaced by Trastuzumab, which is can also be used for the treatment of cancer associated with the stomach. Right. Now, I am going to discuss about the drug repurposing. So now, what is meant by drug repurposing? What was the purpose of the drug previously? I said aspirin. The purpose of aspirin is to produce analgesic activity, right? Now, the purpose of aspirin is it is used for the treatment of myocardial infarction. It can able to reduce the incidences of cardiac complications. That is called the repurposing. Now, the repurposing is very famous nowadays because of the COVID-19. As we know that as per today's statement, there are 9,985 deaths due to the COVID-19 in India, and around 2.76 lakhs of people were suffered for this COVID-19 in India, and all over the world around 73 lakhs people were infected with this COVID SARS-2 virus. Right. Now, as you know that the drug discovery process lasting for 15 years, preclinical, clinical trial 1, 2, 3, post-marketing surveillance, getting permission from the FDA, all these things, right? Animal studies, acute, subacute, subchronic, special time toxicity studies. Now, is it possible to develop the drug for COVID-19? We require the drugs immediately. We cannot wait for 15 years. So what we can do? We can repurpose the existing drug. That's what hydroxychloroquine. We are in a position to supply hydroxychloroquine even for the USA. India is it is a pharmaceutical, rich with the pharmaceutical companies. They can able to satisfy the medicine requirements of the world. So the drug repurposing is by two methods. One is computational approaches. The second one is experimental approaches. The computational approaches are again divided into 6 categories. One is signature matching, molecular docking, genetic association, or pathway mapping, retrospective clinical analysis, novel data sources. Whereas the experimental approaches is binding assays to identify relevant target interactions. Now we are standing on this binding assays to identify relevant target interaction and phenotype screening. Right now, this slide I am showing to explain only the pathophysiology of the SARS-CoV-19. As you know that this is called like projection, all these things, because every day we are seeing in the TV and newspaper. So immediately after fusion entry, this uncoat the genetic material, which is RNA. This RNA can undergo converted into, by the translation, it is converted into polypeptide, the non-structural proteins. The help of RNA-dependent RNA polymerase, there is formation of the RNA and structural protein assembly and release by means of exocytosis. When it is released from the host cell, automatically the virus will, this host cell will get destroyed, so there will be lack of immunity power. So only to boost the immunity, we are taking all the precautionary measures. The government is even government is depends on the alternative system of medicine like Abasara Kurinir and Nilavambu Kashayam, etc. Right now, what is repurposing here? I would like to say now, repurposing approved drugs, it is already approved by FDA, and now WHO is also given permission to do clinical trial of hydroxychloroquine like drugs for the treatment of COVID-19. Right. Now, we can see here, this is the attachment stage. Attachment stage means you have ACE2 enzyme, here ACE2 enzyme, angiotensin converting enzyme, ACE2, or transmembrane protein serine 2, transmembrane protein protease serine 2. These two are very much essential for the fusion of the SARS-CoV-2 with this target receptor of our cell. So now, drug we have selected for the repurposing is nabamostate. Nabamostate is a protease inhibitor. It have antiviral property. It is an anticoagulant. It can also be used for the treatment of cancer. Next one is camostate. Camostate is again protease inhibitor. At the same time, it is also used for the treatment of chronic pancreatitis and also post-operative reflux esophagitis. You have to listen the word post-operative reflux esophagitis. For reflux esophagitis, we can take like cisapride. But for post-operative reflux esophagitis, we are preferring camostate. So these two drugs are now repurposed for the treatment of SARS-CoV-2. The next most important drug is our chloroquine. As you know that the ACE2 are present in the upper respiratory tract, so where it can able to prevent the attachment of this virus to the ACE2. And also, the endosomal acidification is done by means of chloroquine and hydroxychloroquine. Now, if we come to this protease, right, and this is a papain-like protease, and this is chymotrypsin-like protease, and papain-like protease, and these are all the nucleoprotein phosphatase. These can be inhibited by lopinavir, ritonavir. These are protease inhibitor, and disulfiram, which is the most important drug for alcoholic de-addiction. So that drug can be tried for targeting our SARS-CoV-2 because they have inhibitory effect on the papain-like protease. So the proteolysis, right? The most important thing is RNA-dependent RNA polymerase. We know DNA-dependent RNA polymerase. Now this is RNA-dependent RNA polymerase because SARS-CoV is an RNA-containing virus. So that can be inhibited by the RNA-dependent RNA polymerase can be inhibited by remdesivir and favipiravir. So this replication complex can be inhibited. Right now, cyclosporine, which is used for our immunity-related problems, which is acting on the calcineurin pathway. These drugs can also be tried for the treatment of SARS-CoV. So now we can able to say what is repurposing. Repurposing is nothing but the drug is already existing for some purpose. We are repurposing it. So what is the advantage of repurposing? No need of animal studies because the safety is already proven. Phase 1. Phase 1 is on normal human volunteers. That is also over already. Phase 2 is on diseased human volunteers. Now the WHO has given permission to utilize the diseased human volunteers of SARS-CoV so that in near future there may be development of drugs for this particular SARS-CoV. Right. There are other drugs also which is acting on interleukin like Sarilumab and Doctrine. These are also drugs can be utilized for the treatment of SARS-CoV because we are often we used to hear cytokine storm. Cytokine storm, as we know that more than 95 failure antidepressant, anti-cholesterol drugs and asthmatic drugs. So how? Increased by giving the right drug at the right dose to the right patient at the right time. We know that right drug means selection of the drug from the available drugs. Okay, I said earlier, if a new patient is coming with angina pectoris, we have to select either organic nitrates and nitrates, calcium channel blockers, beta blockers, prevention of the cytotoxicity like trimetazidine. How a physician going to select the drug? That depends upon the pathophysiology, right? Right dose, okay. Right time is also okay. What do you mean by right patient? Right patient is his genetic makeup has to be studied. So, a new tool to achieve this objective is our pharmacogenomics. Now, how target should be developed for genetic diseases? Right. If a gene is missing or defective, if the gene is missing, you replace the protein, or if it is defective, you replace the activity. Suppose if any part of our, suppose a bike, if suppose a part is missing, we have to replace the part. Or if the part is no more useful, then we have to replace that spare parts, like that. If the gene is missing, replace the protein. If it is defective, replace the activity, right? If a gene is overexpressing, you have to increase the gene expression. I said Trastuzumab, right? Now, the DNA sequence of all human beings is 99.9% identical, but the 0.1 percentage differences translate into 3 million separate spelling mistakes in a genome of 3 billion bases. That leads to inter-individual variation, right? Now you can see here, I have given 3 types of proteins here. The first is normal. If the gene, the protein is wild type, then there will be normal activity. If there is a excess of amino acid substitution, or if there is alteration in the splice variant, or the truncation of the protein, there will be altered activity. Suppose if the gene is deleted, no activity. Suppose if the gene is duplicate, there will be increased activity. And now what do you have to do in case of gene deletion? We have to substitute that gene. In case of overexpression, gene duplication, we have to suppress the overexpression of the gene. Right. Now, what is the scope of pharmacogenomics? I said only 30 to 60% of the patients are responding for a particular drug. The remaining patients are not responding, or they are responding. That means therapeutic efficacy is not similar. Therapeutic efficacy is not similar for all. So, by means of pharmacogenomics, almost all patients can respond to a particular drug. That is the advantages of pharmacogenomics. Now, here you can see one size fits all drugs. The current approach is one size fits all. Suppose the one size fits all drugs that work only about 60% of the population, whereas in case of individualized approach, that is a personalized medicine, there may be chances of no risk of adverse drug reactions, with the therapeutic enhancement of the therapeutic efficacy by means of the individualized approach, right? And why? What is the problem with this group of subjects? Is nothing but alteration of the nucleotide sequence. That is nothing but single nucleotide polymorphism, right? 75% of the patients in a group, 23% of the patients another group, and 2% of the patients in a separate group. So this explains why succinylcholine induces apnea, right? Why the Premaglin induces hemolytic anemia. So everything can be discussed by using this single nucleotide polymorphism, right? Now, the genomics. What is the use of genomics in cancer drug discovery? Right now, the patient is coming with the sarcoma. Right? What will be the clinical outcome? Either the patient is sensitive for the drug or he may be resistant for that particular drug. Right? If it is sensitive, there won't be any problem. If it is resistant, then the problem starts. So in that case, we have to take the tumoral tissue and we have to study the gene expression profile, and we have to remove the blood sample and we have to see what is the DNA repair capability. So if you can able to say what is the gene expression profile or what is the DNA repair capability, so we can get the 100% success in the therapeutic efficacy, right? Now, because of the pharmacogenomics, there is a cost effectiveness. For example, in case of phase 2 and phase 3, we are going to use only the diseased volunteer. They are patients, diseased human volunteers. And you know, what is the concept of post-marketing surveillance? Post-marketing surveillance, why it has included in the drug discovery process? Because in overall, in drug discovery process, we are utilizing only maximum of 4,000 human population. Either they are diseased volunteers or the healthy volunteers. Maximum of 4,000. But when it comes into the market, millions and billions of people will be using the particular drug. So the adverse effect which is not observed during the clinical trial can able to get the informations from the post-marketing surveillance. So based on the post-marketing surveillance, a number of drugs are withdrawn from the market. That also we know. Right. Now, as per the pharmacogenomics, genetic makeup, we are going to divide the people into two categories. One is responder and non-responder. Doing the clinical trial, carrying out the clinical trial in non-responder is totally meaningless. We have to carry the phase 2 and phase 3 clinical trial only in the responder. So, if you use the genomics, this much amount of the money will be saved during the drug discovery process. Now, what are the benefits of pharmacogenomics? Accurate determination of the drug dosages. I used to give example of amitriptyline. Suppose, with respect to the polymorphism of cytochrome P450, one group of people require 10 mg of amitriptyline, another group of people require 100 milligram of amitriptyline. That is 10 times more dose we have to give. Advanced screening of diseases is possible. There may be chances of development of better vaccines without the risk of conventional vaccine, and improvements in drug discovery and approval process, decrease in overall cost of healthcare, as I have explained in previous slide. Right. Now, other benefits, more powerful medicine based on the proteins, enzymes, RNA molecules associated with genes and diseases are possible. Maximization of the therapeutic effect and reduction in collateral damage is possible. Better, safer drugs for the first time by means of reduction of adverse drug reaction. And these have some barriers also. For example, there will be a complexities in finding the gene variations that affect drug response, whether N-acetyl transferase, etc., etc. Limited drug alternatives. That means the drug will be orphan drug, right? Only one drug is available for that particular disease like that. Whereas if you go for cephalosporin, many number of cephalosporins are available. If you go for omeprazole, many number of omeprazole, lansoprazole, pantoprazole is available. But if you can develop a drug by using pharmacogenomics, there will be limited drug alternatives. Need to re-educate healthcare providers. We ourselves have to be educated about the pharmacogenomics. Right. So this is the pharmacogenomics. This is a conventional method here, right? Phase target identification, phase 1, 2, 3, 4. But what we are going to do here, in case of interpretation of the trials, that is phase 3, based on pharmacogenetic. It is based on pharmacogenetics. And when it comes to the precision therapeutics, it is called the personalization of the drug therapy. Right. Now, this is a drug called PDIL. It is marketed by American Heart Foundation. The company is Nitromed Corporation of American Heart Foundation. They have introduced this particular composition of hydrolazine with isosorbate dinitrate on 23rd June 2005. This is a FDA approved drug. This is the first drug in history to receive FDA approval based on the genomics. And during the clinical trial, they have found that when it was carried out in 1,050 self-identified patients, it was effective only in black people, not with white. There may be chances of the racism, that is called racial tolerance. Right. This is a drawback of pharmacogenomics. Right. The next I'm going to discuss about, I think another 5-10 minutes I will take to complete my lecture. Nandakumar, is it okay? Nandakumar, is it okay? Nandakumar, okay. What are biomarkers in is it okay? Can I discuss by 10 minutes more?
Yes, sir. You can very well proceed, sir. No problem.
Right.
Please, sir.
Now, this is development of biomarkers in drug discovery and development process, right? The biomarkers, in sense, there is advancement in multiple omics. The multiple omics are genomics, transcriptomics, proteomics, cytometrics, and metabolomics. So all these omics are based on the multi-dimensional understanding of the disease biology, followed by the etiology of the particular disease and the pathogenesis of the particular disease. So now, this is possible by 5 different mechanisms. One is predictor, second is safety biomarker, third is mechanistic biomarker, prognostic biomarkers, and surrogate biomarkers. So these 5 biomarkers are employed in drug discovery process. I would like to discuss only about the predictors. For example, vascular epithelium growth factor and fibronectin can be able to predict the clinical response to interleukin-2 in patients with metastatic melanoma or renal cell carcinoma. So, in case of patients with metastatic melanoma or renal cell carcinoma, the biomarkers are vascular endothelium. Please meet your mic. Vascular endothelium growth factor and fibronectin. So, because of the inter-individual variations in drug response, the search for biomarkers for human diseases are now essential, especially for neurodegenerative disorders. Right. Next, I'm going to discuss about the digibiotics. What is, antibiotic, what is meant by digibiotics? Now, please meet your mic, please. Antimicrobial resistance can able to cause 7 lakh deaths per year. Just imagine, because of the development of resistance to antibiotics, there will be 7 lakh deaths in a year. So that it is necessary to develop new antimicrobial compounds with multiple mechanism of action. What is multiple mechanism of action? In case of COVID, we have discussed RNA-dependent RNA polymerase, DNA-dependent DNA polymerase, and supercoiling, that is DNA gyrase inhibitors, etc., or alanine racemase, alanine synthetase. So these are all the enzymes that has to be focused for the development of newer antibiotics. So the aim of digibiotics is to create, develop, and validate the experimental and computational methods for determination of molecular structure, absolute configuration, and confirmation. So this is the latest method of drug discovery called digibiotics. Right. Now, the human genome is made up of polysaccharides, lipids, nucleic acids, and proteins, as we know that. But there is a problem with the toxicity, specificity, and difficulty in creating potent inhibitors and able to eliminate the first 3 categories of things. So, we are mainly concentrating on the proteins, because proteins are called a druggable genome, where you can able to bind the number of drugs with the proteins. So this is the innovations in the drug discovery process. Why protein? It means because most of our enzymes, most of our receptors are made up of protein. So we have to target only the proteins for new drug discovery process. Right. Now lead compound. I think I will take another 5 to 10 minutes. Now, what is lead compounds? Lead compounds, I will say, lead compounds should be efficacy, potency, selectivity. It have physical chemical properties like solubility, stability. It have pharmacokinetic properties like metabolic stability, and that should be easy to synthesize and derivatization. That is very much important. Right. Omeprazole, that is followed by the S-omeprazole and pantoprazole, the rabiprazole. So, because of the chances of the derivatization, the company will get benefit. So, this is the criteria for lead. Right. Suppose if the drug is meant for oral use, definitely the drug should follow the Lipinski rule of 5. The Lipinski rule of 5 is nothing but molecular weight should be less than 500, log p value less than 5, you have less than 5 hydrogen bond donors or less than 10 hydrogen bond acceptor. So a lead molecule, especially for the oral route, should satisfy the Lipinski rule of 5. Right. Now I will give some examples for the lead. This is the structure of sulfonilamide. They have SO2NH2 group. SO2NH2 sulfonilamide. As you know that it is a very good antibacterial activity. Here also you will find the same SO2NH2. Right. This is tolpitamide. Here also SO2NH2, but this is chlorothiazide. So a lead can able to produce different drug molecules. One is an antibacterial, Another one is anti-diabetic. The third one is a diuretic. So, same lead molecules by means of addition, substitution, or deletion of various groups and able to produce the different lead molecules. Here you can see using structural similarity to a natural ligand. We know famous Paravai Kandan, Vimanam Padetan. So here you can see this relationship between 5-hydroxytryptamine and sumatriptan. They are structurally similar. Here, serotonin, 5-hydroxytryptamine, is a very important neurotransmitter. As you know, in case of depression, it is very much involved, antidepressant. Right. Now, if in some cases, amitriptyline was given to a patient, but she is not depressive or psychotic. As you know, that amitriptyline or nortriptyline is an antidepressant drug. But the patient is not psychotic, but amitriptyline was prescribed. You know, amitriptyline, nortriptyline accumulates the 5-hydroxytryptamine in brain, and that is a treatment for the migraine. In migraine, there will be imbalance in the 5-hydroxytryptamine. So, 5-hydroxytryptamine agonists can be used for the treatment of migraine. Right now, identification of the lead compound, I will explain only the serendipity. Serendipity is nothing but it is accidental discovery of new use of an existing drug. The drug is already existing, but you can able to get the new use of that particular drug. I used to give two examples for that. One is phenolphthalein. As a pharmacy graduate or chemistry man, we know phenolphthalein as an indicator, right? But this phenolphthalein was used as a coloring agent in cheap wine. What happened? Whoever consumes the wine which is colored with the phenolphthalein got diarrhea next day. And they have found that the diarrhea is not due to the wine, it is due to the phenolphthalein. So now the phenolphthalein was used as purgatives. And the chemists who are all working with nitrites and nitrates, they have found that severe headache during the night time. So what is the reason for headache? The reason for headache is nothing but the dilation of the cranial blood vessel. The blood vessel which supplies to the cranium is called cranial blood vessel. So the nitrites can able to produce dilation of the cranial blood vessel, thereby to produce a headache. And what the chemist thought, if a drug can able to produce dilation of the cranial blood vessel, why not coronary blood vessel, which is supply blood to heart, right? Now there was development of glyceryl trinitrate, pentaerythritol tetranitrate for the treatment of angina pectoris. So this is serendipity. And some other example I used to give is what is our Viagra. Viagra was a drug sildenafil, was developed for the treatment of hypertension. Now it is used for the treatment of erectile dysfunction, because erectile dysfunction is a disease of obese patient or alcoholic patient. So sildenafil can able to increases blood flow to the male sex organ. Thereby, there was the erection of the male sex organ that leads to the treatment of erectile dysfunction. And this is about serendipity. Right. This I would like to complete my lecture. Before that, this is the new drug development process. Whatever may be the process of drug discovery that has to undergo of all these phenomena, preclinical research, clinical studies like phase 1, 2, 3, and etc. etc. So this I can able to conclude my lecture. If you have any queries, please you can ask.
So thank you, sir, for your
Thank you.
valuable session.
Thank you.
Sir, participants has raised few queries over the chat box, sir.
Please.
So I request you to clarify their doubts. One by one, I will say you the questions, sir.
Definitely.
Mr. Vaspri Rajesh has raised a question. Doctrine of signature, a part of taxonomical classification of plants?
No, it is not taxonomical classification of plants. It is a method introduced by Jacob Binom. It is, I said, it is based on the what we call the color or size, shape, etc. It is not the taxonomical classification.
Thank you, sir. Mrs. Dhanlakshmi has raised a question. What are the limitations in choosing the marine source and how to overcome these limitations?
No, the limitation is only the identification aspect. It is very difficult to identify. As you know, that number of sponges are available, so it is very difficult to what you call the authentication and identification. Of course, the collection of these marine sources is also somewhat difficult.
Dr. Anita Singh has raised a question. Can an hypertensive patient can take aspirin? Is it safe? Please let us know about its effects.
No, if necessary, suppose you have to see the risk versus benefit, right? So suppose if they have a myocardial infarction and hypertension, it is better to take 60 to 75 milligram of aspirin daily, irrespective of our hypertension.
Thank you, sir. Kabita Banik has raised a question. Is there any significance or quality and composition of soil can change the quality of medicinal plant?
Yes, it is possible. No, I am not that much well versed about all these things. Usually, we are adding so many fertilizer only to change the properties of the soil.
Sir, one more question is there, sir. Last question.
Hmm.
Whether whole plant is useful to treat any disease or only the extracting of active materials can effectively treat the disease?
No. Actually, when it is used alternative system medicine, they are using only the whole part of the plant and the extract. They are not isolating that. But when it comes into the market, for example, vincristine and vinplastin from the winter rosea, you have to isolate, otherwise it is not possible.
Sir, one more question has raised, sir.
Please.
Is it possible to come up with pharmacogenomics approach in the present scenario in the nearby future? What are the strategies need to apply and how to update ourselves?
So in newer, there are so many genetic disorders like cystic fibrosis or cancer. For that, you have to go for genomic approach, and especially.
Sir, thank you, sir.
Thank you, ma'am.
Sir, I would like to register heartfelt thanks to the Founder Chancellor, Honorable President, Honorable Secretary, sir, for all their supports in all our efforts. I thank our speaker, Dr. Kavimani, sir, for expecting our invitation and rendering our excellent presentation on the topic, Discovery of Drugs, Doctrine of Signature to Pharmacogenomics. Thank you, sir. I thank all the participants for their support for us. I expect the same energy and effort for all the participants for the tomorrow's session, which starts sharply at 10 a.m. Updates will be given in the groups. Today's assessment link will be processed in the WhatsApp group between 3 to 4 p.m. I request all the participants to take part in the evaluation form. Thank you, one and all.
Thank you.
Sir, thank you very much, sir, for accepting
Welcome.
second time my invitation. Really, this is a special presentation for me. So even for those who are participated in the faculties, so this is very valuable. Thank you once again, sir. Thank you very much.
Thank you. Bye.